Hello, everyone. Today's lecture will be on genetics of prokaryotes, and we've covered this multiple times already. We've talked about prokaryotes, and with genetics, there's not too much to talk about, so this will be kind of a short lecture, but just going into some more details. Again, my introduction along with my email if you ever want to get in contact with me to ask any questions or anything like that. Again, I also recommend joining the GroupMe. We post updates, changes, everything there. It's also an easy way to get hold of me or any of the other tutors, as well as there's a bunch of other students in there. So somebody there will have an answer to your question. Learning objectives. So today we're going to be talking about the properties of prokaryotes, why are they used so often in research. Most research labs actually use prokaryotes, specifically mostly bacteria, for research as model organisms. We also want to know what are the different classifications of prokaryotes. And we'll be talking on something that's kind of related to eukaryotes actually as well, transposons, and do eukaryotes have transposons as well? And how do they work in eukaryotes as opposed to prokaryotes? All right, moving on to the content. First, growth and reproduction. As you remember, prokaryotes are all unicellular organisms such as bacteria. Eukaryotes can be unicellular or multicellular like us, for example. But all prokaryotes are unicellular. As they're unicellular, they mostly reproduce asexually, meaning they go through a process similar to mitosis, which our cells undergo, and that process is called specifically binary fission. In the case of prokaryotes, it's very similar. There are some differences, but I think I covered those differences in a previous prokaryote lecture. Because of this, they often reproduce very frequently because, and some reproduce successfully every 20 minutes. You can turn one bacteria into two into four into eight. And so you can get a colony going really, really quickly because they just reproduce very fast. But because the process of binary fission is fast, the amount of offspring they make is a lot. The process is fast, so it doesn't have a lot of checkpoints and kind of like genetic repair like mitosis does, because they don't really care too much about mutations. They actually end up having a very high mutation rate, and because of this, because they have a lot of mutations, they're much more prone to develop certain aspects or certain genetic variants very quickly in a colony. For example, the most common one is antibiotic resistance, and that's the one we see the most often when we study bacteria. If even one bacteria in an entire colony gets a random mutation which gives it resistance to a specific antibiotic, it'll survive the antibiotic, and then it'll very quickly reproduce, and create a whole colony of bacteria, most of which will share its antibiotic resistance, and it'll essentially make an antibiotic resistant strain of that bacteria. Okay, and again, that's because they don't really have a lot of checkpoints and control compared to mitosis, so they have mutations more often. And as well, when you know, as in cancer, if you just multiply very frequently, you're much more prone, because like 1%, it builds up over time. Okay. Again, one common example is the development of antibiotic resistance. Due to the mutation rate, bacteria will develop resistance to antibody. Once one cell has resistance, it'll begin reproducing very rapidly, leading to a resistant population very soon afterwards. That's the main reason, well, firstly, that it's not recommended for doctors to unnecessarily prescribe a lot of antibiotics to people, because it could create strains of bacteria which are immune to and are like therapeutic antibiotics. And that's why it's also recommended that if you start an antibiotic treatment for an active infection, that you complete the whole course, because even though, say, after the first 3 days you might have killed 80 or 90% of the bacteria, that last 10%, which may be initially resistant to the antibiotic, will begin to reproduce, and the infection will come back. Once they reproduce in the next couple of days, the infection will come back, and this time it'll actually be harder to treat, because those ones that survived had mutations which made it antibiotic resistant, meaning you'll have to try either another antibiotic or a much higher dose of the one you had before in order to fight off the infection. That's why they say you start a treatment, finish it. Just make sure you kill all 100% before they get the chance to reproduce and create a new colony of resistant bacteria. Okay, uses in labs. So similarly to fruit flies, which are the model organism for eukaryotes, bacteria are pretty much the model organism for any labs that aren't specific to eukaryotes. A lot of genetic testing, a lot of DNA stuff and drug testing and stuff done mostly on bacteria. Obviously, it has to be done on eukaryotes and like rats and mice after fruit flies, if you really want to get close to humans, prior to human testing. But this is a good starting point for bacteria. Again, the reasons for that: they reproduce very rapidly. It's easy to manipulate their DNA because it's in a plasmid, and you can just insert it, take it out. And they reproduce very rapidly. So you can have, say, if you put in like one bacteria that's a mutation, you can get a whole colony within a couple of days. It's very easy to study. They're very small size, very compact. So you can have basically entire petri dishes, which have like many colonies, like essentially like on a table. Whereas obviously with bigger animals, like even like rats and mice, you want to have that many specimens, that many organisms, you're going to have to use like a room, right? Which takes up a lot more space, which is something you have to think about when you're running a lab. One way to test a certain gene in bacteria is as follows. So for example, say you have a certain desired gene, a target gene, which we'll call X in this case, and it can be linked to an antibiotic resistant gene. We'll say YR, which stands for like Y resistance. And you put those two genes into a plasmid, and remember they're linked. So if you put in one, you put in the other. If you don't put in one, you don't put in the other. They're linked. So whatever happens to one happens to the other. Multiple of these altered cells are then put into a culture which contains that specific antibiotic that you are resistant against, so Y, and you allow essentially the petri dish to grow for a few days. Any bacteria which do not inherit or do not accept the X gene, which is your target gene, they'll also not inherit or accept the antibiotic resistance gene. Because they don't inherit the antibiotic resistance gene, they will die to the antibiotic, which is in the culture. And anything that doesn't have your target gene will just die. And you know that pretty much everything else that's alive in your culture has the desired target gene, because it also has the linked resistance gene. That's the only reason it survived. Okay. So that's how you can essentially like isolate like a certain gene from bacteria. So just like to draw, here's like a petri dish, has a lot of bacteria on it. They're like dots, and say one of these is the target gene linked to the Y resistance gene, right? And these don't, only this one does. So you put the antibiotic Y into this culture. You let it sit for a few days, and the colony will end up looking like this, kind of. And then when you test all of these will have this X, Y, R, right? And remember, X is what you're looking for. That's because these guys that didn't have X, so they have like, say like a little X and like no YR, I don't know, they didn't have this gene, no resistance, they all died. And this was the only one that was left, and this one reproduced and created a colony. Okay. All right. Next, let's talk about parasitic versus symbiotic. We talked a little bit about symbiotic bacteria in our GI lecture. We have many symbiotic bacteria species living inside our guts because they provide a benefit for us, and they also we provide a benefit for them. So it's a symbiotic relationship. We both get something out of it. It's mutually beneficial. So like I talked about in the GI lecture, they act as protection against harmful bacteria that might invade our stomach, that trigger like diseases, that might trigger diseases. And as well as they provide they also take some of our food that we can't normally digest, and they digest it for us, and we can use some of that energy from that food that they digested for us. And in exchange, they also get a safe environment and ecosystem to grow in, and they get our food, like our access to our food, right? So it's beneficial for both parties, and that's what a symbiotic relationship is. Animals also can have symbiotic relationships. Like one example could be like pets, like us and like cats and dogs. But there's some in the wild, like certain birds and like the crocodiles. If you see like the birds clean the crocodile's teeth, it helps the crocodile because they get their teeth cleaned, and the bird gets food, right? And the crocodile doesn't eat it. So that's a symbiotic relationship. In this case, we're talking about bacteria. So these are like symbiotic bacteria, usually like the good bacteria, what we call them, right? And we want more of these. The opposite would be a parasitic bacteria, which provides no benefit to the host and actually harms the host while benefiting itself. These can have eukaryotic hosts, so some of these bacteria can have us humans or other animals as hosts, and these are usually the ones that cause diseases. So like, for example, salmonella, streptococcus, which causes like strep throat, and these lead to diseases that are very bad for us, and our immune system usually tries to kill these because obviously they're harming us. And meanwhile, there are some prokaryotic parasitic bacteria which can also infect other prokaryotes. That is also something that can happen. I think that's like much less common. I don't think it'll really be tested on it, but just know that it can happen. Most parasitic bacteria are, at least when we talk about MCAT, they'll mention things like salmonella, streptococcus, yeah, like some STDs and other bacteria. Okay. Next, let's talk about conjugation. So remember earlier in the lecture, I mentioned that most bacteria reproduce asexually through binary fission. However, there are ways for them to reproduce sexually, which involves another bacteria rather than just essentially using itself and making a clone of itself. One of these ways is via conjunction. There are a couple of other ways. They're not really tested on the MCAT, so I won't really be going into them. You can look them up if you're interested. The main one is conjugation. This process involves a donor bacteria, which contains a plasmid. And remember, in bacteria, a plasmid is the DNA ring, right? It's like the donut that's has all the genetic information. So there are some bacteria that have plasmids, other bacteria that don't have plasmids. Obviously, if you don't have a plasmid, you can't reproduce genetic information because you don't have genetic information, right? So you have the donor bacteria, which contains a plasmid, and the recipient, which does not have a plasmid. The donor bacteria, which has the plasmid, also has little things sticking out of it called a pileus. And this like little thing sticking out of it, it finds bacteria that don't have a pileus, right? And if they don't have a pileus, that means they also don't have a plasmid. And then it latches onto them, and then the two bacteria kind of create an opening, and they kind of like fuse together, similar to like in mitosis before they split up. They're like two things like fuse like this. Well, it creates an opening in both bacteria. They have like this open semi-state, which I'll show pictures of, right? And then the donor transmits a copy of its plasmid into the recipient, right? It gives some of its DNA into the recipient and kind of throws it in there. Then once the process is complete, the recipient also becomes a donor because now it has its own plasmid, and it also acquires its own pileus, so it can go and be a donor to other recipient bacteria that don't have plasmids, right? And in that way, one bacteria can kind of spread its DNA to others, right? Here's a picture of that. So this is the donor, this is the recipient, this is the pileus. So it's like a little thing sticking out of it, kind of like a USB. And then you can see this one has this plasmid, whereas this one does not have its plasmid. Kind of sticks it in, right? Because it finds it, sticks it in, then it creates like this opening between the two membranes, sends in a copy of its plasmid in here, right? And then they split apart and you have two donors now, both with plasmid, both with pilius. Oh, and this F plus means it has a plasmid. F minus means it does not have plasmid. Right. Okay. Next, let's talk about transposons. So transposons are a stretch of DNA, just like any other gene or DNA sequence that encodes anything else. But the unique thing about this one is that it can change its location in the genome. Literally, physically, it can change its location. So you have a signal sequence surrounding it, which allows a protein called transposase, which is protein's an enzyme, as the name ACE implies. And this transposase finds the DNA, the transposon DNA, picks it up out of the genome that it's in, like picks it up, carries it somewhere else, and then inserts it somewhere else into another area of the genome. There are two types of transposons. There's class 1 and class 2. Class 1 are known as autonomous. Class 2 are known as non-autonomous. Class 1 transposons can move on their own, while class 2 cannot. Meaning that just if you leave a class 1 on its own, it'll activate the transposase and take a transposon and move it somewhere else. It has the ability to do that. Meanwhile, class 2 cannot do that on its own. The reason for that is because it does have the transposon with the signal sequence in its DNA, but it doesn't have a transposase. It doesn't have the protein that actually does the carrying. So what needs to happen to activate a class 2 transposon is you need to be in, you need to essentially, you need to inject a transposase from somewhere else. Usually this will happen if you inject like a class 1 into a class 2. Both the class 1 and the class 2 will then get moved because the class 2 uses the class 1's transposase. Again, it can like borrow it and it'll move the class 2 as well, because it doesn't really differentiate as long as the sequence is attached. Okay. This can also be done in a lab if you just add the protein, like an isolated version of the protein, into a cell. It'll also work, but that doesn't really happen in machine nature. And this is kind of like what it looks like. So this is the transposase gene that makes transposase. Remember, class 2 transposons don't have this. Only class 1 do. So you have this transposon gene surrounded by this sequence right here, these purple things, right? And this blue is the actual gene itself. You have this green protein, which is the transposase, which binds to both sides, essentially like picks it up, takes it with it, and then it ligates it calls for ligation of the original piece, right? Because obviously you're left with a gap. So you have to close the gap, essentially like squish them together. And then it takes it to somewhere else, where like the target, wherever you want to take it, puts it in, inserts it. Okay. So this has some positives and negatives. So these transposons can be used to counter mutations and to increase expression of a gene. So for example, if you want to increase expression of a certain gene, you can just take a copy of the gene and like put it in different spots on the genome. So you can take it and put it in here, put it in here, put it in different spots, and that'll increase expression, especially if you put it next to something that gets expressed a lot. Okay, so like for example, if you were to put it next to something which could be considered, it actually could be considered like an oncogene. If we were talking about humans, if you put it next to something that's highly active, you could end up making a highly active version of this. As well as if you have a mutation nearby which kind of turns it off, you can turn it back on by moving it somewhere else, things like that. So these are a good defense mechanism for bacteria. Bacteria use this to fight off infections and essentially against viruses which might try and change the DNA or something like that, and by shutting off specific genes. However, they can actually be mutagenic themselves, right? So if you land in the middle of another gene where your target is, it can create a gap in the old spot, which is not filled in correctly with ligase. Like I said, it usually like squishes it back together, but sometimes it doesn't, and you can have an error there. You can multiple copies of a transposon in multiple locations could lead to overexpression, which can sometimes be good, but like I mentioned, it can also be like an oncogene, could be bad. You don't want to there's a reason you don't want to overexpress certain genes. Okay, so these have positives and negatives, right? Increased risks, especially in eukaryotes. So most eukaryotes actually do have transposons in their DNA, and this can range from like 5 to like 50% of your DNA could be transposons, depending on the organism. Obviously, humans, I think it's a little bit low on the lower side, but certain have more. But most of these transposons are actually silenced and they're not active. Remember, epigenetically means that the gene is there and the gene normally would be read, but because of a methylation or a deacetylation or something like that, you can actually silence it physically, and you can prevent RNA polymerase from binding to that gene and actually expressing it, which is what silenced epigenetically means. Under certain like stress factors, like in shock conditions, these can actually be unsilenced and activated, but normally you don't want to do that. Normally they're not really activated. So eukaryotes usually have transposons, but usually we don't use them. They're usually just there, inactive. That's why we call them an evolutionary remnant. They are remnant of when we did use them, like way back when we were still like archaea and things like that, and very early life forms, like single cellular life forms. Mostly transposons can be used, eukaryotic transposons can be used for lab purposes, for gene isolation and things like that. Right, if you have a transposon gene, if you just put a transposase, it's really easy to isolate the gene. You don't have to do any like specific cutting, like nuclease stuff. You can just put in the transposase, and the transposase will do the work for you, right? So it's usually used for like lab purposes, eukaryotic transposons. Okay, so like I said, that was a pretty quick lecture. There's not really much to cover. I think we're getting to the point where we've covered the vast majority of the lectures and content, and we're just really going into like the very small details, and like a lot of the stuff is we've already covered in other lectures and stuff like that. So we're just kind of finishing up, kind of wrapping up. Okay, but thank you all for watching. Hopefully some of this was helpful. Have a great time.